Hello everyone, I welcome you all to the 9th lecture of this course. This 9th lecture is on Bio-Nanomachines. So in this lecture we will learn about DNA based nanomachine and the protein based nanomachine. I will also demonstrate a simple experiment to study the motility of bacteria, and we will also learn about the nanomachine communication. So first we will see what is nanomachine. Nanomachines are nano-sized gadgets which doing mechanical work. The examples are like nano gears and nano wheels. The question is, can we make nano motors, and if you want to make nano motors, what are the approaches available? If you see here, there are three approaches: physics based, chemistry and biology based. In physics based approach, we can use carbon nanotubes and we can make nanomachines or nano motors, and in the chemistry based approach we can use the supramolecular chemistry and we can make a small nano size machine, also nano size switches. Okay. And in the biology based approach we can use the DNA and protein for making various nano size machine or rotor. Okay. So how to construct this nano machine? The first step for constructing nano machine is modeling and design, so which comes under nano architectonics, and these are the fields contributing to this modeling and design. And for synthesis of nanomaterials, mainly the chemistry, biochemistry, these fields contributes in the synthesis of nanomaterials. And the next step is fabrication. So for fabrication of nano devices we need the contribution from this cell biology and other fields, and finally you have to integrate and make the complete nano machine. Okay. So what would be the application of this nano machine? Let us see an example. So we can make a drug delivery nanobot, that is nano robots, so which can precisely reach the tumor location and it can release the anti-cancer drug. So here you can see here it is specifically binding to the cancer cells, and also it is releasing the drug, and we can make such kind of small size nano machines not only for drug delivery application and also for various other applications. So we can take the idea from the cell, okay, because the cell is the most sophisticated existing nano machine, and it has a efficient energy consumption and it has also multitask computing and multi sensing capacity. So when the cell, we can easily take the idea and we can mimic such kind of nanoscale machines or nanoscale robots. So you can see this slide, it is a complex metabolism. So this kind of metabolism is going on in your cell every milliseconds or nanoseconds, and without any disturbance or without any trafficking inside the cell. So how is it possible? And when we understand this kind of complex things, and we can make a very small size nanoscale machine which can do the job very precisely and which will have wide applications. So that is why we are taking the idea from the nature, so we can make the biologically inspired nano machines. So how to make the nano machine, what are the approaches? So there are again two approaches, bottom up and top down approach. So man made nano machines are top down. So we started with robots and we are trying to make nano robots, and but nature it started with bottom up approach. So it assembles DNA and protein and makes the nanoscale machines, and especially in case of nano machine the DNA and protein play a major role, because the manipulation of DNA or hybridization will allow us to obtain user defined biological nanomachine. That means like we can easily manipulate or we can easily modify the DNA and we can make such kind of small nanomachines, and also we can make a user defined biological nanomachine. So that is the advantage of DNA and protein based nanomachine. So in the previous lecture I already told you that A always form bond with T and G always form bond with C, that is cytosine. Okay. And also I already explained why the DNA is helical in nature, what is the reason for the helical structure of DNA. So let us see the various forms of DNA. So here we have three forms of DNA, that is aDNA, bDNA and zDNA. So the main difference, if you see that aDNA have 11 base pairs per turn of helix, and bDNA have 10 base pairs per helix, and zDNA have 12 base pairs per helix. So these are the various forms of DNA, aDNA, bDNA and zDNA. So using this, how to make nano machines and nano motors we will see in the following slides. So before we see how to make nano machines, let us get idea about some of the terminologies. So here you can see here this is a double helical region, and this B is a sticky ends, that means overhangs, and C is your bulgy loop, and D is the hairpin loop. If we have this kind of DNA sequence, that is called as hairpin loop, and this E is the junction and this F is the crossover. And using this DNA, we can stretch the DNA, we can condense the DNA, and also we can play with the DNA by using external fields. So before we learn what is DNA based nanomachine, so let us learn what is protein synthesis. So we will get the RNA from the DNA by the process transcription, and from RNA you will get the protein by translation process. So we can take the example of transcription and translation, we can make such kind of small nano machines. So let us see translation. So you can see here, this is your mRNA and this is your tRNA, and this orange color is your rRNA. So the tRNA is coming and binding to mRNA, and another tRNA is coming. If it is not matching, it will be removed. So this tRNA is matching with the sequence, so it is forming a peptide bond. So similarly, one by one amino acids will come and join and form the protein. So you can see here how precisely it is moving. So it is also a kind of nano machine, and at the end of the reaction you can see here this mRNA is getting degraded. So this poly A tail is very important. If the length of poly A tail is more, that means your mRNA half life will be more. Okay. So assume this is as a kind of nano machine, and we can take the idea from this and we can make such kind of nano machine. So let us see the another example, how to make a DNA based rotor or motor. Okay. So here we can use the DNA and we can use that RNA polymerase enzyme. So what happens is, like when it synthesizes RNA, the DNA will start rotating in this direction. So that we can easily measure by tagging the DNA with this magnetic bead, and the top of the magnetic bead we can also add the fluorescence beads, and this magnetic bead will be held by external magnetic field. So when we add this RNA polymerase enzyme, so this will make the RNA. When it makes the RNA, the DNA will rotate in this direction. So this rotatory motion can be easily monitored using the microscope. So the next example is DNA based nanomachines, and in this we can see like various machines are available, like B is the transition, molecular tweezer, and also PX and JX2. So these are the various types of DNA machines available. So we will see one by one. The first one is B is that rotor. So this machine is based on transition between bDNA and zDNA by changing the ionic strength of the medium. Okay. So this motion can be easily monitored by FRET. So what is FRET? It is a mechanism describing the energy transfer between two light sensitive molecules. So here we can simply changing the ionic condition of the medium. So we can convert the bDNA into zDNA. So this is your fluorescence molecule and this is your quencher. So when it is in the bDNA form, it will not show any fluorescence. When you change the ionic condition, ionic medium, so it will become a zDNA. So where your fluorescence molecule as well as quencher will be separated, and it will give the fluorescence. So here you can see here, when it is in the bDNA form, both the fluorescence and quencher will be together. So when you change the ionic condition, so this bDNA will become zDNA, and your fluorescence and quencher will be separated. So when it gives, then it gives fluorescence. Okay. So this is called as B is that transition. So by simply changing the ionic condition from bDNA, we can make the zDNA. In the bDNA, the fluorescence and quenchers are together, so it will not show any fluorescence signal. So when it becomes zDNA, when it becomes zDNA, what happens is that the fluorescence and quencher will be released and it will give the fluorescence signal. So the next example is rotatory DNA nanomachine. So here this device works by producing two different conformation, parallel conformation and zigzag conformation, by using a two pairs of strands, that is called as set strands. Okay. So that will binds to the this DNA single stranded, and it will make this kind of parallel or zigzag conformation. So here you can see the example. So this double stranded DNA, it is holding this single stranded, and when we add this kind of set strands, and it will form this kind of parallel conformation and zigzag conformation, depends on the size of your set strands. If the set strands matching with this size of your single stranded DNA, it will produce parallel conformation. If it is slightly bigger than the your single stranded DNA, it will form zigzag conformation. So here your DNA is acting like your fuel. Based on the DNA, it will have a rotatory motion. Next example is walking triangle. You can have this kind of DNA sequence, and when you add a short fragment of DNA, single stranded DNA, the red color one, it is binding to this single stranded DNA in between these two triangles, and there are also some unbound the DNA. So that DNA will form like a loop like structure. So in this case, both the triangles will be on the top, and when we add the red color strand DNA, which is matching with the single stranded strand, that is the blue color DNA. So when it forms a hybridization, what happens is this triangle will rotate, it will have 180-degree rotation and it will move to the bottom. So this is also another example for DNA based nanomechanical device. So the another example is DNA tweezers. Okay. So this is your DNA tweezer. Here you can have fluorophore, that is your tetrachlorofluorescence, in short it is TET, and another one is quencher, that is TAMRA. This is called as tetramethyl rhodamine, in short it is called as TAMRA. So using this we can make the DNA tweezer, we can open the tweezer and we can close the tweezer. So what we can do is we can add a single stranded DNA. So this blue color will bind here and this green color will bind here, and it forms this kind of structure. When it forms this kind of structure, this fluorescence and quencher are close together. So when it is close together, the fluorescence signal will be off, and when we add another sequence which is highly specific for this, the blue color and green color strand, so it will combine and this strand will be removed. So when it removed, this will be in a open state. So here you will get the fluorescence, because the fluorescence and quenchers are separated, so it will give a fluorescence signal. So in this picture you can see here the fluorescence intensity is more when it is open state, and the fluorescence intensity is down when it is in the closed state. So same DNA tweezer in animation you can see here, when the strand binds it became closed state, and when the strands get removed again it became the open state. So let us see the another example, that is a protein based nanomachine. So that is dynein, F1 ATPase and bacteria flagella. Okay. So here this dynein is a molecular motor that walk along the microtubule in a cell, and ATPase synthesize ATP by using influx of protons to rotate, and bacterium it use the flagella to move from one location to other location towards the chemical, that is food. So first we will see bacterial chemotaxis. So this bacteria move from one location to other location using the flagella, that means it move towards the food. Okay. And it get the signal from the food and it move towards that food, that is called as chemotaxis. It get the chemical signal and the bacteria move towards the food, that is called as chemotaxis. So here the bacteria move using flagellar motors, and this protein network directs movement based on the external conditions. If you have food, that is attractant, or if we have some other material, that is repellent. Okay. And this will simulate chemotaxis network. It will simulate more than 7 proteins, then only this flagella will be in action to move towards the food. So here the bacteria move towards chemical attractants. So the process is the attractants bind to the chemoreceptors, and the chemoreceptors transmit information to a central processing system, and the central processing system integrates many inputs and sends a signal to control the flagellar motors. So the bacterial motility will be look like very simple, but you can see that how much reactions are going on to make the bacteria to move from one location to other location. So what we can take from this bacteria, we can take the sensitivity. So if we have any food materials, it is sensing and moving towards that. So we can take the idea of sensitivity and we can develop such kind of nanomachines and nanorobots which can sense the cancer cell in our body, and it can reach the cancer location and it can deliver the anti-cancer drug. Here you can see here, so if we have sugar solution, all the bacteria is moving towards the sugar solution. So and bacteria swim by rotating the flagella and motor located at junction of the flagellum, and this motor can rotate clockwise as well as counterclockwise. So this bacteria swims smoothly for 1 second and it change the direction by an average of 60 degree. So, movement is with respect to the attractants. If we have increasing concentration, it will be less tumbling, and if we have decreasing concentration, it will have more tumbling. You can see here the bacteria flagella, it is like a kind of nano machine or nano motor. So, now I will demonstrate a simple experiment to study the motility of bacteria. By using hanging drop method, we can study the motility of bacteria. For this, we need a concave glass slide. So, this glass slide will be having concavity, okay, and we also need a cover slip. So, on the 4 sides you will be adding a Vaseline, okay. So, keep a simple dot of Vaseline on the 4 edges. Then in the middle of the cover slip you add 5 to 10 microliter of bacteria. So, here we are going to add GFP E. coli. So, GFP is green fluorescent protein expressing E. coli. So, by using this bacteria we can easily monitor the motility of bacteria. So, once we add this bacteria to this cover slip, then gently keep this glass slide on the top of this, and invert the slide. So, when you invert the slide, so your cover slip will be on the top of this concave slide and your bacterial solution will be hanging. That is why it is called as hanging drop method. And under the microscope you can study the motility of bacteria. So, let me explain the hanging drop method to study the motility of bacteria. So, for this we need a concave slide and cover slip and also freshly grown bacteria, but in this case I am not going to use the bacteria, I am going to use the simple water, okay. So, this is your concave slide, okay, and this is a cover slip. So, on the 4 edges of cover slip I am going to put the Vaseline. Just put one dot on all the 4 corners. So, all the 4 edges I have added the Vaseline, okay. So, I hope it is visible, and in the middle of the cover slip I will be adding the sample, okay. So, we have to use the bacteria which is in the log phase, so that you can see the bacterial motility nicely, but in this case I am adding water just to demonstrate you. Yes, for bacterial sample you have to use the laminar hood and you have to prepare the sample aseptically. So, then I will take this concave glass slide and put it on the top of this cover slip, then gently I touch this cover slip. So, you can see here, so the cover slip is in the middle of the concave glass slide and your sample is hanging in the middle of the cover slip. Slide I will keep it under this microscope and you can focus the motility of bacteria. In this slide you can see here we have recorded the GFP E. coli bacteria motility. So, you can from this slide you can understand the bacterial motility. So, let us see another example, ATP synthase, okay. So, here the proton gradient drives F1 rotation accompanied by ATP synthesis from ADP. So, if you high ATP concentration that drives rotation in opposite direction with ATP hydrolysis which pumps protons. Okay. So, here you can see here during ATP synthesis it will rotate in the clockwise rotation, and during ATP hydrolysis it will rotate in the counterclockwise rotation. So, it is a kind of reversible motor, and here the F1 unit was attached with the diloaded actin and attached to F0, and you can easily see the movement of gold bead by laser optical imaging. This is the gold bead, you can see here rotatory motion, and here the motor takes 3 120 degree steps to complete one rotation. That means it takes 3 120 degree steps to complete one rotation, and also hydrolyzing 1 ATP molecule per step. Okay. So, now let us see nano machine communication. So, why this nano machine communication is important? So, the nano machines such as chemical sensors, nano valves and nano switches, this cannot execute the complete task by themselves. So, the exchange of information and commands between network nano machines will allow them to work in a cooperative and synchronous manner to perform more complex tasks such as in the body, drug delivery as well as and disease treatment applications. Okay. So, the nano machine communication is very important if you want to make a successful nano device or nano motor for drug delivery applications. So, let us see in case of biological nano machines, we can make this kind of logic gates. Suppose if both substrate and effector is present, then only it will make the product. If there is no effector or no substrate, the substrate remains unchanged. So, we can make such kind of logical gates. For example, if your cancer cell is expressing two kind of receptors or two kind of markers, so then only this nanoparticle will bind, the nano machine will bind and it will release the drug. Okay. So, it will not release the drug to any healthy cells. So, we can target it for various drug delivery applications. So, in this molecular communication, here the sender and receiver both are biological nano machine, and the communication carrier, the molecule, that is also in the range of nanoscale, proteins, ions and DNA, these are in the range of nanoscale, and the communication distance, it will be become nano or microscale, and the receiver which receives the signal, that is also in the range of nanoscale. You can see here the sender is a nano machine and also the receiver is also nano machine, and the information which is going from sender to receiver, that is also nanoscale, that is protein or ions or DNA. So, here the molecular communication is divided into two types, that is molecular motor, that is a wired, and another one is calcium ion based, that is wireless. You can simply assume that this molecular motor is similar to your telephone connection, landline connection, and this calcium ion based wireless is your similar to your cellular phone. Okay. So, let us see the communication using molecular motors. So, first we will see what is a molecular motor. So, it is a protein or protein complex that transforms chemical energy into mechanical work at a molecular scale, and it has the ability to move the molecules. Here this is your microtubules and this is a molecular motor, okay, and it is the information. So, this molecular motor is carrying the information from one location to other location through the microtubules. So, it is mainly found in the eukaryotic cells, okay. So, it can carry the message and it can walk on the microtubule. So, the molecular motors move along the molecular rails called microtubules, and the movement created by molecular motors can be used to transport information molecules. So, here you can see here this nano machines communicate using these molecular motors, and these molecules are transported from encoder to decoder using this network of rail. Okay, it is this molecular motor will walk on this network of rail and it reach from encoder to decoder and it will carry the information. So, here how the communication happen in these molecular motors, because it select the right molecules that represent the right information and it will carry the information, and through the microtubules it will move and it reach the receptor, okay, and there your molecule will be detached and it will release the molecules. Okay. So, once the receiver receive that information, it will perform the work according to the information. So, the next example is communication using calcium signaling. Okay. So, here two different deployment scenarios are available. The first one is direct access and another one is indirect access. So, here the exchange of information among cells located next to each other, and in indirect access cells it can be separated without any physical contact and the information can reach the receiver. So, let us see the direct access, that is your gap junctions. So, all the cells are connected and there is a nanoscale protein channels between the two adjacent cells. So, it will allow the small molecules to be shared among the cells and it will make the coordinated action of the cells. Okay. So, in direct access the calcium signal travel through the gates, that is your gap junction. From here the information it will go to the decoder receiver through the gap junction of the cells. Okay. This is the direct access example. Next one is indirect access. Here the nanomachine release the information molecules to the medium. So, the calcium molecules can go to the receiver which is little far away, okay, and the receiver will receive the information and it will perform the task according to the information. So, here the information is encoded in calcium and it will involve the signaling initiation, and also it propagates the calcium waves and receiver receive the calcium concentration. So, according to the calcium concentration the receiver perform the task. So, what is the main application of this? We can have the pinpoint drug delivery, we can deliver the drug to the targeted cancer cells very specifically. So, as I told you earlier, if both substrate and effector is there, then only it will form the product. But still lot of challenges needs to be addressed, because we want the system to be autonomous, we do not want any human control, and we want the system to be closed, that means no energy supply from outside, and we also want the recycling of the carrier in molecules, and again we want slow and sustained delivery of drug molecules. So, as a summary of this lecture, we have learnt what is DNA based nanomachine and protein based nanomachine, and we have also learnt nanoscale communication, and this field need a lot of research to explore the potential application of nanomachines in various fields. Okay. So, I will end my lecture here. I thank you all for listening to this lecture. I will see you in another interesting lecture.